Okay, so today we're doing chapter 9. This chapter involves cellular respiration and how we harvest chemical energy. So 9 and 10 are going to be linked chapters, so we will spend some time talking about how they are linked. But for chapter 9, we are talking about how this process occurs in animals. So this is specific to animals in the way that they harvest their chemical energy through the processes of cellular respiration. There are a couple of key concepts that we're going to talk about in this chapter. So we're going to talk about both aerobic respiration and anaerobic respiration. So when we talk about aerobic respiration, we are talking about processes that utilize oxygen. And towards the end of this chapter, we'll talk about anaerobic respiration, and those are processes that do not utilize oxygen. Now, aerobic respiration occurs in 3 phases. We're going to talk about glycolysis, we're going to talk about the Krebs cycle or the citric acid cycle, and we're going to talk about electron transfer phosphorylation. As I said, photosynthesis, which is chapter 10, and aerobic respiration are linked sort of on this global scale. So when we think about the way that they work, 2 of the byproducts of aerobic respiration are carbon dioxide and water. So when we carry out aerobic respiration, we give off carbon dioxide and water. Plants then use our carbon dioxide along with sunlight to carry out photosynthesis. So this process up here occurs in plants or other photosynthetic organisms. The byproducts of photosynthesis, so when plants and photosynthetic organisms carry out photosynthesis, they give off glucose, or they produce glucose. That's what they use as their food source. But they give off oxygen, and that's the key to the linkage, because we utilize the oxygen to carry out aerobic respiration. So you can see this is a linked process between aerobic respiration and photosynthesis. And we'll be talking about the ways that they're linked in the throughout this chapter and the next chapter. All right, so the whole process of carrying out cellular respiration is so that we can capture the energy of glucose and convert that energy to ATP. ATP, remember, is your cell's energy currency. So that basically represents energy inside your cell. So what we do is we take glucose, which is in the food that we eat. It's a sugar, it's a carbohydrate, and through the processes of cellular respiration, we're going to break that glucose down into ATP. This process starts with glycolysis, which occurs in the cytoplasm. So glycolysis is step 1 of this process, and it occurs in the cytoplasm. Once we harvest the energy that we can from glucose through glycolysis, we're going to continue if we're an aerobic respirator. If we are utilizing oxygen, we would move into the mitochondria, where we're going to carry out the citric acid cycle and what we call electron transfer. So we'll talk about those 2 steps in a second. So those processes occur in the mitochondria. If we are an anaerobic respirator, meaning that we do not use oxygen, we cannot move to the mitochondria and carry out the citric acid cycle and electron transport chain. These processes absolutely require oxygen. And so if we are an anaerobic respirator, if we do not use oxygen, we cannot carry out those reactions. So our processes, if we're an anaerobic respirator, end in the cytoplasm. We cannot move on to the mitochondria. Organisms like humans and other organisms that can use oxygen can carry out the citric acid cycle and electron transfer chain in the mitochondria. All right, so let's look at a quick overview and comparing aerobic respirators from anaerobic respirators and looking at the energy sources. So in aerobic respirators, these are again your air breathers, where anaerobic respirators are mostly your bacteria and your yeast. So things that we use to make bread, yogurt, wine, some cheeses, all of those things are what we refer to as anaerobic respirators. So they do not require oxygen, whereas our air breathers, like humans and other animals, absolutely do require oxygen. So if we look at the phases, remember glycolysis was the first phase of respiration, and what this actually means is that we're going to split glucose. We're going to lyse or break apart glucose. Both aerobic respirators and anaerobic respirators carry out glycolysis. So glycolysis happens in both types of organisms. In the locations, we know that glycolysis starts in the cytoplasm, and we know that anaerobic respirators can then move on to the mitochondria, where they carry out the citric acid cycle and the electron transport chain. Anaerobic respirators cannot. They stay in the cytoplasm. They cannot move on to the mitochondria. So anaerobic respirators, that's one big difference between aerobic and anaerobic respirators. Anaerobic cannot carry out the citric acid cycle or the electron transport chain. Therefore, their processes end in the cytoplasm where they begin. We'll talk a little bit later on about what this means, the final electron acceptor. But in aerobic respirators, that final electron acceptor is going to be oxygen. In anaerobic respirators, the final electron acceptor is going to be a molecule called pyruvate, and we'll talk about that in just a second. So then another big difference refers to how many ATPs we get per one molecule of glucose. So in aerobic respirators, when we take in one molecule of glucose, we can produce about 36 ATP molecules. So that's a good amount of energy for us for every one molecule of glucose. In anaerobic respirators, they can only make 2 molecules of ATP for one molecule of glucose. So they're making much less energy per glucose molecule as the aerobic respirators are. Okay, so let's do a quick overview of aerobic respiration. So again, here we are. We're going to take in glucose through our diet or through other sources that we acquire glucose. We're going to break that glucose down in the process of glycolysis. In glycolysis, we're going to make 2 ATP. We're going to net 2 ATP, and I'm going to explain to you in a second what that means that we net 2 ATP. We also are going to make 2 molecules of this enzyme helper called NADH, and we'll talk about where that's used in just a second. And then sort of the final product of glycolysis is these 2 molecules of pyruvate, and we'll talk about sort of how we get there in just one second, but that's sort of the overview of glycolysis. We're going to break down that glucose into 2 molecules of pyruvate. Those 2 molecules of pyruvate then can go through what I like to call the prep step. So we'll talk about that in a second. I'll call that the prep step. And then it can enter the Krebs cycle or the citric acid cycle. So this is a cyclical metabolic pathway in the cell that is responsible for making some energy for us. So you can see we get 2 more ATP here out of the Krebs cycle. We also give off some CO2. Remember, plants are going to use that to carry out their photosynthesis. So they're going to take up that CO2. We also now get 6 more molecules of NADH and 2 more molecules of another enzyme helper called FADH2. By the way, let me go back in the prep step. So that's this step right here. We also give off 2 additional molecules of CO2, and we get 2 additional molecules of NADH. So all of these guys, the 2 NADH from glycolysis, the 2 NADH from the prep step, the 6 NADH from the citric acid cycle, and the 2 FADH2 from the citric acid cycle, all of those things go down here to be utilized in what's called the electron transport phosphorylation. That's where we make the bulk of our ATP. See, we get 32 ATP from that process, which is most of our energy source here. And this process utilizes that oxygen as what we call the final electron acceptor. So you can see we yield about 36 ATPs per 1 molecule of glucose that we started with up here. So that's a quick overview of aerobic respiration. We're going to take a closer look at these steps as we go through the chapter. All right, so here we are in glycolysis. We start with a 6 carbon sugar, glucose. So these little brown circles represent carbons. So you can see glucose has 6 carbons that we start out with here. And you're not going to be responsible for the names of the molecules or specifics of these steps, but there are a couple of things that I want you to know. So in this first step, we're going to take glucose and we're going to phosphorylate it to glucose 6-phosphate. So this little yellow circle here, that's a phosphate group. So in order to do that, we have to use 1 molecule of ATP. Now I just told you the whole purpose of this process is to make ATP. And here we are in the first step utilizing a molecule of ATP. So it's kind of like that old expression, it takes money to make money. It's kind of the same thing here in the cell. It takes energy to make energy. We have to input a little bit of energy to make this whole process go, to make this metabolic pathway flow. And in fact, we have to use 2 molecules of ATP. So you see in the first 3 steps, we have utilized 2 ATP molecules in order to set this process in motion. So here we are, we've got glucose, we phosphorylate it to glucose 6-phosphate, we change that to fructose 6-phosphate, we add another phosphate group here to make it fructose 1,6-bisphosphate. All of these molecules have 6 carbons. You can see all the little brown circles represent carbons. And then in this last step, we do something kind of interesting. We split that molecule in half, right down the midline here. So we take that 6 carbon molecule and we now have 2 3 carbon molecules. So everything that we do from here on out in glycolysis, in the citric acid cycle, and in through glycolysis, the prep step, and the citric acid cycle happens twice for each of these molecules of what we call PGA. So that's going to be important to remember, that all the processes that we talk about from here on, the rest of glycolysis, the prep step, and the citric acid cycle happen twice so that we can take care of both of these molecules of PGA. All right, so here we are with the rest of glycolysis. Remember, this is a metabolic pathway, and so we took those 2 molecules of PGA that we had at the end of the last slide, and we now convert them to these other molecules called PGAL. So we go on down through the process, but now if you look here in these steps, we have produced 1 molecule of NADH. So here's, so now we have 2 NADH molecules that have been produced in that step. We now have in the next step, you can see that we dephosphorylate something. So here we have this molecule 1,3-bisphosphoglycerate, and here we have 3-phosphoglycerate. So we took off that phosphate group, we removed it, and what we did is we used it to make ATP, and so we made 2 of those because we made another one right here. So we make 2 molecules of ATP, and then down here we did the same thing. We have this phosphoenol pyruvate that has a phosphate group here, and we take it off and we transfer that phosphate group to make some more ATP. And again, we make 2, so we make 1 here and we make 1 here. So now we've made 2 more molecules of ATP. So far in glycolysis, we have made 2 molecules of NADH and 4 molecules of ATP. But remember, we had to use 2 molecules of ATP to get this thing started in the last slide. So kind of 2 of these cancel out. So this is what we used to start it. We've made 4. But because we had to input 2, we really only netted 2 ATP. And we'll talk more about this on the next slide. The final product of glycolysis is this molecule called pyruvate. This is what's going to enter the prep step and eventually enter the citric acid cycle. So remember, we get 2 of those. Okay, so the second stage of anaerobic respiration is the Krebs cycle, or the citric acid cycle, and this is where pyruvate is going to enter the mitochondria. So that pyruvate that we just made from glycolysis is now going to, remember that happened in the cytoplasm, and so now pyruvate is going to move to the mitochondria so that it can enter the Krebs cycle. Remember, the mitochondria is a double membrane organelle, so it has two membranes. It's got the outer membrane out here, and then it has this sort of curvy inner membrane. So two membranes surround the mitochondria, and the Krebs cycle happens in this inner compartment, the innermost compartment inside the inner membrane. All right, so before we can enter the citric acid cycle or the Krebs cycle, we have to carry out what's called the prep step. We have to prep pyruvate to enter that cycle because it can't enter as pyruvate. It has to enter as something called acetyl-CoA. So pyruvate is going to be stripped of a carbon atom. We're going to take one carbon atom off, and we're going to convert that to carbon dioxide. So remember, plants are going to use that carbon dioxide to carry out photosynthesis. Pyruvate is also going to give up a hydrogen and some electrons to form another molecule of NADH. So we'll talk a little bit more about that. And then coenzyme A is going to join with the remaining two carbon fragment to form this molecule acetyl-CoA. Acetyl-CoA is what can directly enter the citric acid cycle. So we're going to look at a picture of this now. Okay, so here we are. We have our, we're in the inner membrane of the mitochondria, so this is where the prep step and the Krebs cycle occur. We have pyruvate here with its 3 carbons, and remember we have 2 of these from glycolysis. We made 2 pyruvate molecules, so this is happening twice, even though I'm only showing it to you once. So we have our 3-carbon pyruvate molecule. That molecule is going to, first of all, lose one of its carbons to form carbon dioxide. So that's going to be given off. That's a molecule that plants are going to use in order to carry out photosynthesis. We're going to take some electrons and some hydrogen ions off of pyruvate to make some NADH. And so remember, we're going to make 2 NADHs because we have 2 pyruvate molecules. We're also going to make 2 CO2s because, again, we have 2 pyruvate molecules. And then you're going to have this molecule called coenzyme A that's going to come in and join with the 2 carbons that are left, and that produces this molecule called acetyl-CoA. Acetyl-CoA is what can actually enter the Krebs cycle and be utilized to make some more ATP. Okay, so let's talk a little bit about the Krebs cycle then. Acetyl-CoA transfers its 2-carbon fragment to oxaloacetate to start the cycle. So that is the first reaction of the Krebs cycle. So acetyl-CoA is going to join with this other molecule called oxaloacetate, and that starts the cycle off. During the cycle, we're going to make some more NADH, we're going to make some more FADH2, we're going to make some more ATP, we are going to make some carbon dioxide, again what the plants are going to use, and this is a cyclical cycle. So the last step of this cycle regenerates oxaloacetate so that the cycle can go repeat itself all over and over and over and over again. So we'll talk a little bit about that. Okay, so here we are in talking about the prep step, or talking about the citric acid cycle. Again, you do not have to know the all of the steps of these cycles and all the names of these molecules, but I do want you to know what the outcomes are, what the products are. So here we are, this is our prep step, where we have pyruvate going to acetyl-CoA. And so again, we've made 2 molecules of NADH, 2 molecules of CO2, and 2 molecules of acetyl-CoA throughout this process because we had 2 molecules of pyruvate from glycolysis. So the first thing that happens is acetyl-CoA is going to join with this molecule called oxaloacetate, or OAA, and it's going to produce this molecule called citric acid, and that is how the cycle gets its name, why we call it the citric acid cycle, because that's one of the products, one of the first products of the cycle. So as you can see, as we go around the circle, as we go through the cycle, we're going to make 2 molecules of NADH, we're going to make 2 more molecules of NADH, we're going to make 2 molecules of ATP, 2 molecules of FADH2, and 2 more molecules of NADH. So in total, we make 6 molecules of NADH, 2 molecules of CO2, 2 molecules of ATP, and 2 molecules of FADH2. This is important. So this is what I want you to know from the citric acid cycle, is what these outcomes are, or what the products are. All right, so once we get through the citric acid cycle, we need to enter the electron transport phosphorylation. So again, this only occurs in aerobic organisms. Anaerobic organisms cannot carry out the citric acid cycle or the electron transport chain. So here we are. And remember, this is where we make the bulk of our ATP. So we're gonna make 32 ATPs here. Remember, we have only netted so far. We netted 2 molecules of ATP from glycolysis and 2 molecules of ATP from the citric acid cycle. So far we only have 4 molecules of ATP that we have made. All right, so the electron transport chain occurs again along that inner membrane of the mitochondria. So remember, we have the outer membrane and the inner membrane. So we are along that inner membrane of the mitochondria. We're going to be dependent upon hydrogen ion concentration gradients, as well as electrical gradients set up by other ions along this membrane system. We have a special enzyme that we're going to use here called ATP synthase, and I'll talk about that. And this is where we are going to form the bulk, remember, of our ATP. So we'll talk about how that happens. Okay, so the first thing that happens is we have made all of that NADH and FADH2 from glycolysis and the citric acid cycle. So we've made all of those molecules. Those guys, their job, they are electron and hydrogen ion taxis, okay? Electron and hydrogen ion taxis in the cell. So their job is to deliver the electrons and the hydrogen ions to the transport chain. So here in the inner membrane of the mitochondria, these guys are bringing their electrons and their hydrogen ions and sort of dropping them off here. The hydrogen ions are going to be shuttled from the inner compartment to the outer compartment. So every time an electron bumps down this chain, so here's our electron, and it's going to move from molecule to molecule to molecule, kind of down this chain. You can think about it as a ball falling down a set of steps. So this electron is going to bounce and jump down this chain from one molecule to molecule. As it does that, these hydrogen ions are pumped out to the outer compartment of the mitochondria. So we're going to set up this concentration gradient out here with all these hydrogen ions in the outer compartment. So the gradient that we're going to set up is going to be high in the outer compartment, low in the inner compartment. So that's going to be really important. Okay, so the electron, remember, is going to bump down this chain. It's going to fall down the steps until it gets to the last step. And the thing that is waiting for it at the end of that step, at that last step, is oxygen. Remember, this is why we called oxygen the final electron acceptor. Okay, so that electron's going to get down here to that last step, and oxygen is going to be waiting to scoop it up. So we now have oxygen, we have an electron, and we have a little bit of hydrogen ion concentration in here. Remember, this was low out here, and this was high out here. So we got a little bit of hydrogen ion concentration. When we take hydrogen, oxygen, and an electron and put those things together, we make water. Okay, so remember, that was one of our big products of aerobic respiration. Water was a huge product. So remember, our products, when we take glucose, and we're going to break those things down, plus oxygen, because we're aerobic respirators, we take glucose plus some oxygen, and we break those things down, we're going to make CO2 and water. So we've been making CO2 all this time in the citric acid cycle and in the prep step, and now we've made water. So here is our product of water that we get here in the electron transport phosphorylation. So oxygen is used as that final electron acceptor to keep that electron moving through the chain and to keep the hydrogen ion concentration low in this inner compartment. So we cannot have hydrogen ion building up in this inner compartment. So one of the ways that we do that, we get rid of it two ways. Number one, we pump it out, right? So every time an electron flows through the chain or flows down the steps, hydrogen is pumped to the outer compartment. The other way that we keep it low is we use it to make water. So we use it in this process here to make water. So we don't let that hydrogen ion concentration build up inside that inner compartment. Okay, so we have our hydrogen ion concentration here, and they sort of flipped the picture on us here. So now remember, this is the inner compartment. This is where our hydrogen ion concentration is low, and out here is the outer compartment. So this is where our hydrogen ion concentration is high. So we have this concentration gradient set up. We know, remember, that ions move from an area of high concentration to an area of low concentration based on gradients and the way that things flow. So what happens is we have this special enzyme here on the membrane. So this enzyme is called ATP synthase. And so what this enzyme does, it's responsible for making ATP. The way that it does that is when a hydrogen ion flows through this protein or this enzyme, a hydrogen ion concentration, because remember, diffusion is going to happen. We are going to diffuse from an area of high concentration to an area of low concentration. So hydrogen ions are going to flow through this enzyme to sort of even out the concentration gradient. So as they flow through, they cause this reaction to take place. ADP plus a phosphate. So we have adenosine diphosphate plus an additional phosphate yields ATP, adenosine triphosphate. Okay, so remember, that was the whole point of what we're trying to do here, is make ATP. And in fact, we get so much oxygen out here in the outer compartment that flows through this enzyme that we end up making 32 molecules of ATP. We get 32 of this reaction happening so that we produce those 32 molecules of ATP. All right, so we spent a lot of time talking about aerobic respiration and what happens in oxygen breathers. I want to talk now a little bit about anaerobic respiration. So these are organisms that do not use oxygen. So one of the ways that they can produce energy and keep themselves alive is through alcoholic fermentation. This is one type of anaerobic respiration. This is performed by yeast as well as some bacteria. This is the process that causes our bread to rise when we're making bread. It's because of the CO2 that's produced. You get those CO2 bubbles that are made, and that's what causes the bread to rise. It's also how we make some alcoholic beverages, is through alcoholic fermentation. So what happens here is we carry out glycolysis just like we do in aerobic respiration. So this is glycolysis just like we've been talking about. We start with glucose. We're going to break it down into 2 molecules of pyruvate. Along the way, we're going to make 2 molecules of NADH. We're going to make 4 molecules of ATP, but remember we had to put 2 in. That's our net. So, I'm sorry, this is our input. So we had to input 2 molecules of ATP to get this thing started. We made 4, so we net 2. All right, it's sort of like if you made, if you had a yard sale and you made $100, but you had to spend $25 to buy all your supplies for the yard sale, you only netted $75. You only came out positive $75. So that's what happens here. We had to put in 2 ATP to get the reaction going. We made 4, but our net, what we actually gained, was only 2 ATP. So that's true of glycolysis for aerobic and anaerobic respiration. It's exactly the same. The difference is when we get down here to alcoholic fermentation, we take that pyruvate and we remove one of those carbons just like we did in the prep step to give us CO2. But now we're left with what's called acetaldehyde. We don't have that acetyl-CoA that's formed here in sort of this prep step. We now have something called acetaldehyde, which is going to join with the electrons and the hydrogen ions from NADH to form ethanol. So ethanol is sort of the final product of alcoholic fermentation. And obviously you guys know that we use that to make things like beer and wine. So there's our ethanol that we get from alcoholic fermentation through an anaerobic process. The other anaerobic process that we can talk about is lactate fermentation. So this is also anaerobic, again, utilized in organisms or in situations where oxygen is low. So we use this in milk, we use this to produce cheese and yogurt and sauerkraut, lots of things that we like to eat. What's interesting is that humans also carry out lactate fermentation during periods of high endurance exercise. So if you've ever been working out and your legs kind of start to burn, you start to feel that burning sensation in your legs, that is because the cells in your muscles are carrying out lactate fermentation. They don't have enough oxygen to carry out the citric acid cycle or the electron transport chain, so they have resorted to anaerobic conditions where they are carrying out lactate fermentation. So this is what your muscle cells use to make ATP when oxygen is depleted. So that burning sensation is actually a buildup of lactate or lactic acid inside your muscle cells. So when that molecule builds up through this process of lactate fermentation, you start to experience that burning sensation. When you return to normal oxygen levels, so when you stop exercising and you start taking in enough oxygen, that lactate breaks down inside your cell, and so that's why the burning sensation sort of goes away. All right, so here we are with lactate fermentation. So again, glycolysis is exactly the same that we've been talking about. Here's our 2-net ATP. The difference here is those two molecules of pyruvate are converted directly to this molecule lactate, again, through the combination of the electrons and the hydrogen ions from NADH. So this is what builds up inside your muscle cells, and this is what causes that burning sensation. So that is the end of chapter 9. I will see you in class.